Hello, everybody, and welcome to another exciting episode of Animal Physiology. It's always amazed me how we run on this invisible gas that we just kind of walk through in our environment. So, today we're going to talk about respiration. And respiration is quite a process. And I think of respiration, I think of species like this. This is a tiger salamander, both the adult form as well as the pedomorphic form, which lives in the water. And I feel amphibians kind of highlight how incredible the process is because they make that transition from water to air and they shift from having to extract oxygen and get rid of their carbon dioxide in water, and they're able to essentially seamlessly start doing this in air. So today we're going to talk about the processes that are involved here. But first, let's talk about how respiration differs between water and between something that an air breather would need. So first, let's start by looking at some of these differences at sort of a sort of chemical level. So right off the bat, you can see that there are some differences in these numbers across these different variables with regards to water and air. Some of the notable things that stick out is that water has a very low concentration of oxygen compared to that in air. And in air, it's about 21%, as you probably have learned, right? Where it's more like 0.7% in water. So that's that first little row right there. So that's a 30-fold difference between air and water. Some other things you might notice when you look at the diffusion coefficients, you might notice there's a big difference between oxygen in air and oxygen in the water, and vice versa for carbon dioxide. And we'll get to that a little bit later. Some other things you might notice is that there's a big difference in the weight that you need at the bottom there, kilograms of medium per liter oxygen. In order to get so much oxygen, 1 liter's worth, you need 143 kilograms of water, whereas you don't even need 1 kilogram, not even 1% of a kilogram to get that in air. So there's a very big difference, and you can start to see the implication of coming onto land. And one of the reasons we evolved to come onto land, which is to gain access to oxygen in our environment, which would have been especially important in the Carboniferous when vertebrates first started making their forays onto land, which was thought to have been a relatively low oxygen time. So moving on from this table, we could start to see the relationship between chemistry and physics and respiration. It's important to remember that we are extracting a gas from our environment, oxygen, and we are releasing a gas into our environment, carbon dioxide. So it makes sense that the laws that govern the movement of gases are also going to govern and put an upper and lower limit on what we can do as a species. So the respiration is going to be driven by these laws of gas diffusion. So, for example, we know air is made up of a variety of gases, and each of these exert their own partial pressure. And when you look at the percentages of gases, you notice that oxygen is not the most common gas, nitrogen is. But oxygen is still pretty common, whereas carbon dioxide is much, much lower. Now, as gases are going to move from one place to another, they're going to do so according to their own molecules, their own partial pressures. So even if a gas is under high pressure, that's a mixture, individual gases and individual elements are going to move according to their own dynamics, i.e. their own partial pressures. But again, something like air, which is a gas mixture, is going to have an overall pressure that's going to be made up of partial pressures of individual components of that gas mixture. So we can model all this using basic chemistry. So PV equals nRT. I think some of you might have right away picked up on the fact that this is the ideal gas law. So pressure times volume equals nRT. N is number of moles. R is this interesting number, 8 1.314 joules per moles per K, per Kelvin, degrees Kelvin, and T is temperature. So together, this allows us to make predictions, really accurate predictions, about how pressure is going to move from one place to another to drive diffusion, which again, when we're talking about diffusion, we're talking about the movement of gases across some sort of biological membrane, whether that's the lining of the lung, the the capillaries and tissues where they meet, or it could be the gills, right, where it meets the water. So a quick thing, this R right here, this is the ideal gas constant. It's the constant that you need to make these predictions, right? So it's just a number that's been verified over and over again as being able to accurately predict the movement of these gases. So we're really good at understanding how gases move from one place to another. Now, this is all based on air, and these are the components of air, and shown in both their volume fraction, i.e. the percentage they make up, but also in a molar fraction, right? Which is either way, you're looking at the partial pressure. And taken together, we like to use this unit of atmosphere. So at sea level, the pressure that all of these gases exert are going to be about this. Now, this is in air. Now, what about aquatic environments? Well, aquatic environments are a little more complicated. So as temperature increases in an environment, the solubility of gases in that liquid decreases. So this is a way to essentially release gases, is often by manipulating the temperature. So here's a question that involves around these two graphs here. And what these graphs show is temperature on the x-axis and oxygen solubility on the y-axis. And they're showing two. The one above is for fresh water, the one below is for salt water. And you have three different colored lines. And basically, the blue line is under a low pressure of oxygen, right? Partial pressure. The orange obviously is in the middle, and then the yellow is under a high pressure of oxygen, right? So this is saying that there's higher pressures of oxygen in the air above a water interface. So what do you see here? Well, you see that all these six of these lines on these graphs show general decreases with temperature. So that's kind of what you predict. Solubility goes down. But you also have these differences among bars. So one's in fresh water, one's in salt water. So now here's the question. What does this mean for the animals trying to live in these environments? So you have two options. Which one do you think? Take a quick look at the options you have. Okay, so your two options are basically asking which is more efficient in extracting oxygen from the water. Is it going to be a saltwater creature or a freshwater creature? Well, when you look at the graphs right away, you notice that there is a little bit of a difference between the two graphs in terms of where those lines fall. And in freshwater, you seem to have a much higher solubility of oxygen, which means that there's probably higher levels of dissolved oxygen in freshwater as compared to saltwater. Ergo, if you have this difference, then you would expect that creatures living in the saltwater are going to be a little bit better at getting oxygen out of their environment, all else being equal, simply because there's less of it to have. And assuming that they have the similar oxygen demand as a freshwater animal, so for example, like a freshwater fish versus a saltwater fish, similar oxygen demand, then it means that the saltwater creature is going to have to be much better at getting it out of its environment. So that's just sort of a basic explanation of how respiration is responding to differences in the environment based on some sort of chemical or physical property. So another thing to remember is organisms are exposed to high amounts of oxygen in their environment. 21% of the molecules floating around you right now are oxygen molecules, are O2, exactly what you need. And yet, we're always constantly in need of oxygen, right? We never store it. We never just have it, right? We constantly need it. And that's because even though we have oxygen all around us, by the time you get to where you need oxygen the most, the mitochondria, in order to power aerobic respiration, by the time you get down there, that availability of oxygen has been depleted with every step of the way. It's a law of diminishing returns. So here, this is known as the oxygen cascade. So in human beings, well, you have a high partial pressure in the air of oxygen, but as soon as you breathe it into your lungs, you right away decrease it by almost a third, right, in alveolar gas. So this is the gas in the alveoli, which we'll talk about later, that are essentially available to diffuse into your bloodstream. And by the time you get into the bloodstream, well, there isn't much of a change there. But then by the time that arterial blood makes its way all the way to the capillary beds where it's going to diffuse out into the cells that need it, well, right off the bat, you're almost at half the amount of oxygen that you started with or had available to you. And by the time you get down to the mitochondrial level, you're looking at very, very low partial pressures of oxygen. So what does this mean? Well, this means that you can power your body off of very little oxygen, really. But it also means that you have to be very, very good at extracting oxygen from your environment. Now, there's a number of ways that animals have learned to do this, quote unquote learned. They haven't really learned. Evolution provided answers to this problem. Um, give you a hint, it's basically one of the few answers that your body does have for things like this. But this sets up a situation where oxygen demand is a constant thing. So when we talk about diffusion, there are actually very few creatures that can rely on this entirely. So this is an earthworm. If you know anything about earthworms, you know that earthworms are very slow. You know that earthworms don't move around much. Earthworms are very soft and have a very permeable membrane, and they have, they're very small, so they can actually have a respiratory system where they're basically just relying on gas diffusion itself, because diffusion is essentially limited to about a millimeter in distance. That's about how far gas can diffuse effectively across a membrane or a respiratory exchange membrane, right? Now, this does put limits, because if you think about an earthworm, well, what is one of the big things about earthworms that happens to them, especially if they're out in the hot sun? They dry out. In order to maintain gas diffusion, they have to maintain a wet membrane all the time, which is why they're very susceptible to water loss, because they can very easily desiccate completely and lose all of their water because they have a very permeable membrane. But they can survive in environments where they have a relatively low metabolism anyway. They can get their oxygen directly from their environment, which is often very low concentrations anyway, if you think about underground. Now, this is an invertebrate, and there are many others that do this, but there are some vertebrates that can use diffusion. Can you name one? Well, I think of what I showed you earlier, amphibians, right? There's a lot of amphibians that can do respiratory exchange across their skin, so cutaneous respiration. Frogs, some salamanders, stuff like that. So gas diffusion has its place, but simple gas diffusion can be tricky. One group that has done a really good job of using differences in partial pressures to drive respiration through a simple diffusion mechanism are insects. And the way they do this is they've compartmentalized air, meaning that they basically have a way to get air into their bodies, and then as air enters their bodies naturally through the differences in partial pressure, they're able to get it to the cells that they need. And they do this by having these openings on the side of the body called spiracles. And what happens is these spiracles will open up, air will rush in, and then we'll go through a network of trachea. And the trachea are kind of like our lungs. We have a trachea too. That's our windpipe. But then our windpipe branches into bronchi and bronchioles and gets down to very small tubes. And in the end of an insect, they break down into tracheoles. And tracheoles are kind of everywhere, and they completely cover the insect, so that you can actually have tracheoles be within 1 millimeter of the cells of any cell. So what that means is any cell can get air just through diffusion through this network of tracheoles. And you can actually visualize this if you do X-ray crystallography, which they've done here in this bottom image. They use like an X-ray image, and you can see all of these tubes running inside. The other thing some insects have, especially insects that tend to fly and have high metabolic rates anyway for an insect, is they'll have air sacs, which help to kind of store the air temporarily and release it slowly, right? So this is showing kind of how diffusion is working in grasshopper. Grasshopper is breathing in, that's the yellow. It fills up these air sacs, and then it breathes out through another path, which is another big thing that they'll do. Is insects will basically keep the flow of air in one direction, meaning that they're not going to breathe in and out of the same spot. They're going to breathe in through some of these spiracles and breathe out through other spiracles. They'll breathe in high oxygen air and release CO2 later somewhere else. And this is known as discontinuous gas exchange. They've decoupled the breathing in of oxygen with the breathing out of CO2. So that's how they get oxygen to their cells. That's how they get carbon dioxide out of their cells and out of their bodies. Um, Now, insects also have a blood equivalent known as hemolymph. Is there a role for hemolymph in this, you think? Well, the answer is no. So hemolymph has like a greenish substance. It's got a lot of copper in it, but it's not quite like our blood because our blood is separated from all the other fluid. And we just call all that other fluid, we just call it intracellular fluid or interstitial fluid. And then blood is something else. In the case of insects and other invertebrates, there is no distinction like that. Their circulatory systems are open. So hemolymph is just floating around and it's indistinguishable from interstitial fluid. And even though there are these colored pigments with copper that give it this greenish tinge, that's not there to be involved in respiration. So unlike us, where we have blood and our blood is red because of hemoglobin, and our hemoglobin is there to bind oxygen and help to move it around, which we'll talk about. In the case of insects, they don't have that. They don't need that, right? They have this other system, so they don't need to transport oxygen or carbon dioxide with the pigments like we do. We need hemoglobin to do that. So it's not involved at all. Okay, so the next question is how do we move air in and out? And it's important to understand the distinction between respiration and ventilation. Respiration is the use of oxygen, the production of carbon dioxide at the cellular level, and it's the use of these things to essentially provide fuel and energy. All right, so aerobic respiration, mitochondria, all that, transport chain and all that. Ventilation, on the other hand, is a term used to describe the physical movement of air or water across a surface. So when we talk about breathing, that's the physical movement of air in, out of your body. So that's the physical movement. That's ventilation. That's what breathing is. Respiration occurs when the oxygen that's in your lungs now is going to make its way into the blood. It's crossed a barrier, and as soon as it crosses some kind of a membrane, it's involved in diffusion, it's involved in respiration, and that's what that term refers to. So big difference between ventilation and respiration. So physical movement, ventilation. So as far as evolution goes, there's basically two ways that animals do this. They either exhibit tidal flow, which is like the lung basically, just like our lungs. You breathe in air, you breathe out air. You breathe in air, you breathe out air. You breathe in air, you breathe out air. And it's going in and out of a sac that inflates, deflates, inflates, deflates. So in the case of tidal flow, you're going in and out of one opening. Whereas convective flow is unidirectional, meaning that you are basically moving through your environment, or the environment is moving through you, and you're engaging in respiration exchange at some point in this process. So the best way to think of this is tidal flow is lungs, and anything that resembles unidirectional convective flow, that's basically gills. So gills are just kind of hanging out in their environment, and diffusion is happening. If you're a fish like a shark, you're moving through your ocean, and that the movement of water past your gills, that gas diffusion is happening across the respiratory exchange membrane. You're not necessarily bringing it in. I mean, you could bring it into your mouth and then pump it out of your gills, right? And your gill slits, that happens too. Fish do that. It's called buccal pumping. And we'll talk about that. But the bottom line is that these are two very different ways to do ventilation. So we tend to think of lungs as being this thing that air breathers have, and gills being this thing that water breathers have. But there is a creature out there, quite an interesting creature, that actually does ventilation with lungs underwater. It has water lungs. It's crazy, right? Water lungs, right? And despite the fact that it's rare, a sea cucumber has a pair of respiratory trees that are inside it. And these respiratory trees are connected to the cloaca. So the cloaca is an opening. It's the single opening to the environment of a sea cucumber. And basically what it'll do is it'll bring in water in through the cloaca, fill the respiratory trees, and then it'll contract, and then it'll breathe out the water just like a tidal lung, right? So the respiratory trees are basically like lungs. And during this time, the sea cucumber is going to extract whatever oxygen it can from the water, right? So what type of lung is this? Well, it's a tidal lung. It is a water lung, basically. But here's the thing: why didn't selection favor this type of water lung? Why don't we see it more often? Why is it only found in sea cucumbers? And the answer lies in kind of how sea cucumbers are. Well, if you know anything about sea cucumbers, I'm guessing you've never been to a sea cucumber race before. But if you did, that was probably a very boring time, because sea cucumbers, frankly, do not do shit. They don't do anything at all. They sit there breathing, and it's kind of it, right? And they'll accumulate water, and they'll expel water and accumulate water and expel water. When I was a kid, I used to grab sea cucumbers, and I used to chase my sister around, squirting her like a water gun with sea cucumbers. Now that I'm older, I know that that water was actually the water that was in the respiratory trees in these guys. So the reason they don't do a lot is because this method requires a lot of energy. You are moving water. You're not moving air. Think about how much energy it takes to move some air out of your lungs. You could breathe out very easily. Now think about how much energy it would be to expel water, right? So there's the physical movement of a substance that is much heavier, much more viscous, right? And then on top of that, it's water. So there's much less oxygen in there anyway. So you have to set up a very elaborate system to encourage water in the oxygen, the dissolved oxygen, to make its way across into the fluid inside your body, which is also oxygen. There's not as big a partial pressure difference there. So the fact that it's very hard to move and requires a lot of energy, it's a big energy investment for sea cucumbers, and it's part of the reason why you've probably never been to a sea cucumber race, because they're spending all their energy moving this stuff around. Right? And then also, you're not really getting much benefit from it because there isn't a lot of DO in there in the first place. So, great idea, nice experiment. Sea cucumbers are pretty beautiful, especially when they got their full colors, but not the best respiration system. So, moving on up, let's talk about gills. So, gills can be either, exhibiting either passive ventilation or active ventilation. In the case of passive ventilation, what they're doing is they're relying on the movement of water or the movement of themselves to encourage, brand new oxygen-filled water to go over their gills, right? So something like a lamprey, for example, it's showing here the gills on a lamprey. Lamprey aren't very fast, but what they are good at is sticking to things. So they'll attach themselves somewhere, and usually they're living in streams and brooks, and they can just kind of attach, and they can allow the movement of water down the river, basically providing oxygen for them, right? So as the water goes over their gills, they'll extract what they can, and then the next little bit of water is going to have a higher amount of oxygen, and they'll be able to extract that again. And so they're constantly just pulling out of their environment with fresh water because the water is moving around them. In contrast, something like a shark will do something where it's swimming around. You've probably heard that sharks need to keep swimming or they die. That's not necessarily true. There are sharks that don't do that, but there's a lot of sharks that do need to keep moving around. And what they'll do is they'll rely on ram ventilation, where they're basically moving quickly through their environment, and with their mouth open, water rushes in through their mouth and rushes out through their gills. All right, it's because they're moving, and it's a good system because the faster you move, the faster water moves over your gills, the more oxygen you get, right? So it's a good system where you're coupling fast movement with more oxygen, which you need to do the fast movement. So it's like a bidirectional thing. So these are both passive, even though they involve movement. There's not something that's like a specific mechanism that involves your own energy. In contrast, other fish like teleost fish, which is most fish out there, it's fish with gills basically. If I ask you to impersonate a fish, right? Look in the mirror, impersonate a fish, what would you do? Well, you'd probably start opening your mouth, opening, closing your mouth, like, and you'd probably also put your hands up to your just under your ears and start pretending like you have gills and you'd open and close them, right? So that's basically buccal pumping. So buccal means cheek. So they're basically just producing a pumping thing. Kind of like the sharks, they open and close their mouth. Every time they open their mouth, a fresh batch of water moves in, and then every time they close their gills or they do muscle contractions on something called an operculum, the gill cover, and every time they do that, they force water out. So they're bringing in a new batch of water with brand new oxygen and low CO2 content, rush it over their gills. They pick up what O2 they can, dump what CO2 they can, and then they get rid of the water right away. So that's buccal pumping. So that's a form of active ventilation because it requires muscle contractions, which requires ATP. You know the drill. So in water, this works great. Now, I told you how the water is moving. How do you think blood flow in the gills, which again are just capillaries really, how is blood flow in the gills going to vary with respect to the movement of water? Think, this is physiology after all. Well, hopefully you said it's going to move in the opposite direction. Counter current exchange. It's a great simple system that allows you to maintain a constant gradient by having things move opposite to each other, and it encourages the movement of things back and forth. So in a situation where you had concurrent exchange, where water and oxygen would be moving in the same direction, diffusion across the lamella, which is the term used to describe where gas diffusion is happening, the membrane, the membrane that, where the gills end and the environment begins. What would happen is, well, you'd have a very high partial pressure, which is shown here in these pictures as these numbers here. Partial pressure, the relative partial pressure of oxygen in the water is 100. So in the gills it'd be 0. It's never that, but whatever. You would get a very big gradient, and soon they would equilibrate. And once they equilibrate, there's no more real gas exchange, so you're not really getting anything anymore, right? So the concurrent exchange would give you a lot, but not much, and it wouldn't be sustained. Whereas if you're doing countercurrent exchange, now water is moving in one direction, gills are moving the opposite direction. Well, that means that all along the lamella, you're going to have a constant difference in partial pressure that's going to force oxygen from the water to constantly go into the gills. And it won't be as dramatic, but it will be more sustained. And that's important because that means that if you need more, evolutionary speaking, you can just make more gills and more gills, and you have more and more, right? So you'll able to constantly have exchange. So the more lamella you have, the more you can exchange. The bigger your gills are, the more oxygen you can exchange, right? Whereas the other concurrent version, you would basically have the same amount, regardless of how long your lamella are, for example. So this is important because countercurrent exchange, surface area, those are the rules of physiology in terms of what you can evolve. Okay, so that pretty much concludes water, and I think it makes a lot of sense. What about air? At some point, one of our long distant ancestors decided that air is better. So what are the benefits? Well, there's more oxygen in the air than in water, so there's a benefit right there, right? You have access to more oxygen. Partial pressure is higher in air than in water. And because it's air, and air is not very viscous, and it's lightweight and easy to move, then you could have a tidal flow in a situation like this. So something like a sea cucumber, struggles in the water, but that same sort of system on land can work, right? And we know this because we do it. But there are some costs as well. So if you look at this picture here, it's showing blood and the movement of blood, and those numbers indicate oxygen partial pressures. And it shows a tidal lung. And in the inhalation, it absorbs, you're breathing in, inhaling, and you're bringing in 100%, I guess, partial pressure of oxygen, whatever that partial pressure is, 21%. You're breathing it in. And right away, you see that number go down to 60, right? And then you breathe out 60. So why did that number go down? Well, some of it went down because it quickly got across into the blood, right? And we extracted it. But there's another reason too. And that's because you will never completely get rid of the air in your lungs, right? When you get rid of the air in your lungs completely, like every bit of air, that's basically a medical situation, a collapsed lung, right? That's what it means. So you're always having to maintain a small residual amount of air in your lungs just to provide its shape, and because you just can't get rid of it anyway, even if you wanted to. And this is often called the residual tidal volume, which is the volume in the lungs, or dead space, right? Anatomical dead space. So what this means is that you're always going to have this old, stale air inside the deepest recesses of your lung, and you're going to mix that old stale air with the brand new fresh air that's got all the oxygen that you want. So that's kind of a problem, right? Because right off the bat, you're kind of lowering your efficiency by having this stale air in there. But you need it, but it still lowers your efficiency. The other risk you have is, unlike something living in the water, you have to be concerned about losing water. So water loss, evaporative water loss is a big thing. That's especially problematic for species living in dry environments where water quickly escapes the lung, and it's a major contributor to sort of desiccation, drying out. Desert animals have to be concerned about water loss. They often adjust their behavior to coming out at nighttime just for, having less water loss. Anytime your internal environment is exposed to the atmosphere, you can lose water. The other issue is on the carbon dioxide side. When you breathe out, you're breathing out carbon dioxide. But there's a problem, because what you're breathing out is going to be in much higher concentration than what you're breathing in, which is good. But there's also a problem in that your lungs are going to have a very high partial pressure of CO2. But we already talked about how your CO2 doesn't even like to be in a gaseous state and would prefer to be soluble in blood. So what that means is that you sort of have to find a way to force CO2 out of your blood and into the air, because it would rather be dissolved in your bloodstream. So this sets up an issue, because especially if you have dead space, then that stuff can tend to accumulate CO2 pretty quick, and you have to find yourself having to force CO2 into, where there's a very low gradient. So there's benefits to air breathing, of course, but there are some costs as well. So this sets up a big issue. Air breathers get more oxygen and have no problem doing that, but they have a hard time getting rid of their CO2. So they load oxygen easily, unload oxygen very difficultly. On the flip side, things breathing in water, they have a hard time getting O2 from their environment, but they have no issue getting rid of their CO2 because it likes to dissolve, right? So oxygen doesn't like to be in water, and yet we're forcing it into water. CO2 likes to be in water, and yet we're forcing it into air. So this sets up a big problem. So once again, we can look at amphibians. So amphibians make this wonderful example of respiration because depending on where you are in life, they're having different challenges. So during the tadpole stage, well, they're trying to extract oxygen from their environment, which is often very low concentrations, very low partial pressure, because it doesn't dissolve very well. But they have no problem getting rid of CO2 because CO2 is more soluble. Now, as they transition from being a tadpole living exclusively in the water to being an amphibian that is now able to take advantage of the air, well, they get this interesting situation where they can basically take advantage of the best of both worlds. So they can breathe in, and they have a lung. They'll breathe in air that has oxygen, so they'll get the oxygen demand. But they'll have this issue, like the rest of us, where CO2 is harder to get rid of in the air. And this is often why you see frogs, for example, basically putting half of their body in the water, just kind of sticking out on the shoreline with half their body in the water. Well, at the base of their pelvis, they can actually undergo cutaneous respiration. And what they'll often do is they'll breathe in oxygen very easily, both their lungs as well as through their skin. But then by being in the water, they're able to get rid of CO2. So they're actually able to expel CO2. So they have the best of both worlds, right? They can breathe in the oxygen, which is floating freely in the air, and there's so much of it, and it's wonderful. And by hanging out in the water, they can also just get rid of CO2 through their skin, right? And it becomes less of an issue for them. They don't have to worry about some of the problems that we'll get to, right? So they get the best of both worlds, which I'm not saying I want to be a frog, but in terms of respiration, you could do a lot worse, right? Think of that sea cucumber again. Now, for land breathers and us lung organisms, you see this really clear relationship between the surface area of the lungs that allows for respiration, as well as this relationship between sort of overall metabolism and respiration. You see this big shift when you go from being something that is, ectothermic or heterothermic, like say a salamander or a lizard, to becoming an endotherm, like a bird or a mammal, right? And you see sort of an increasing emphasis on being able to draw in more air and have more respiratory exchange membranes to be able to do that. By the time you get to the birds, their lung is not necessarily much bigger, but they have a network of other, structures, air sacs, that will allow them to essentially optimize respiration. So when we talk about lungs, and we'll use the mammalian lung as the example, the human lung, the one that's got all the pictures on the internet you can grab. You have, obviously you're breathing in through your nasal cavity as well as you can breathe in through your oral cavity. All that stuff eventually goes down your windpipe or your trachea, right? And then goes down the trachea and then splits into two main bronchi, primary bronchi, that then split into secondary bronchi and tertiary bronchi and eventually become bronchioles. The difference between them is that trachea and the bronchi have cartilaginous rings in the form of a C. And the reason that they have a C ring as opposed to a complete ring that surrounds the entire trachea, the reason is because right behind the trachea you have your esophagus, right? And every time you swallow something, that thing swells up. And what you want is to be able to accommodate the esophagus swelling up, but not completely close off your airway either. So C rings allow for that a little bit of give as the esophagus swallows something, but also maintains the shape that allows it to not completely collapse and cut your air off. As you go down the trachea into tracheoles, etc., you'll eventually, or bronchi, bronchioles, sorry, just like an insect there for a second, you'll eventually terminate on the alveoli. Now the bronchioles are called bronchioles because by that point you have no C rings, it's just smooth muscle. And they can contract and relax just like other types of smooth muscle. But at the terminal end of these things, you have where the gas diffusion occurs, which is the alveoli. And the alveoli, they look kind of like grapes on a stalk. Yeah, they are what they are. And you have many of them. And you have so many of them that it basically meets your needs. And by the time you get to the alveoli, they're surrounded by capillary beds, and you're able to have gas diffusion because you have very thin alveolar membranes that are less than 1 millimeter, much less than 1 millimeter. So you have a lot of them, you have a lot of capillaries, a lot of diffusions happening. You can't control that part because that part is happening in still air. Dead space is part of that. So you're going to get the diffusion you're going to get. What you can manipulate, however, is the actual amount of air you're bringing in. So lung volume will vary, right? You'll have this normal lung capacity, and we use the term capacity to describe the volume and the ability to bring in stuff, right? So we have a tidal volume that is your normal. But then, as you know, if you have to, you can breathe in more, you can breathe out more, you can manipulate this, right? So this is just kind of showing changes in lung volume and the different terms we use to describe, breathing patterns, right? So we talk about, you have your normal tidal volume, but then you also have a reserve volume. This is a reserve volume that you can breathe in a lot more, right? If you need to. If you take your deepest breath, right, you can basically expand your inspiratory capacity, and then your expiratory capacity as well, how much you can breathe out. And the total range of what you can do, and everybody has a different, that's called your vital capacity. How much can you breathe in? How much can you breathe out? That is called your vital capacity. So these are different terms. They mean relatively little. Note that a chunk of it is something you can never get rid of. There is a volume you can never get rid of, right, which is about 1 liter. That's your residual volume, right? So you're never getting rid of that air. That's the still air that mixes with your fresh air and lowers your partial pressure of oxygen. It is what it is. So when you start to look at it, you realize that most of your lungs is involved in moving the air, the ventilation part. And the respiration part, the exchange surface, is very small. But when you actually look at it as like a surface area, it becomes quite something, right? Because you're basically talking about, somewhere along the lines of, 100,000 or so centimeters squared in terms of surface area available for a single breath from which oxygen can do gas exchange. So that's pretty impressive, right? So even though your lungs are relatively small, there's a lot of surface area embedded in them. So it goes to show just how intricate they can be, right, with all these alveoli. And there's kind of an interesting thing to think about. You forget sometimes how small a lot of things are. But if you have enough small things, they can amount to a huge amount, right? So this is an example of the respiratory tree. So it's the same thing basically the same sort of thing that the sea cucumber had. So we're like a glorified air-breathing sea cucumber as far as I'm concerned. So let's talk a little more about these alveoli. I already mentioned that they're one cell layer thick. There are basically two types of cells. Here you have type 1, type 2, go figure, alveolar cells, why not? They are slightly different in how they look. Simple squamous epithelium, all that means is that they're basically kind of flat looking, right? And this is where the gas exchange is happening. And these are the majority of your cells that make up the surface area. And there's less than a micron distance, or roughly about a micron distance, between the alveoli and the blood. So there's very little sort of membrane that gas would have to diffuse across, right? Whether that's CO2 or O2. And then you have these other sort of interspersed type 2 cells that their main job is to produce surfactant. And if you know what a surfactant is, then you know that a surfactant, usually we think of it as this thing that breaks apart lipids and stuff like that. So think about like detergents, for example, or soaps are surfactants. But what they really do is they basically decrease tension, surface tension, and allow gas to move through a little easier. So it actually helps to sort of, break up some of the hydrogen bonds and to make sort of polar bonds a little less polar, allowing for substances to move through, right? So it decreases surface tension. And by doing so, it basically allows that gas diffusion to occur a little easier, right? So a lot's going on inside your lung, and you don't even pay attention. Shame on you. All right, so your lungs themselves, you've got a right lung, you've got a left lung, multiple lobes, 2 lobes on the left side, 3 lobes on the right side. They're encased in pleural membranes. There's an outer parietal layer and an inner visceral layer, right? So we call these pleural membranes, pleura. These are general terms. Parietal, visceral. Usually parietal describes the outside membrane. Visceral describes the inside membrane. You see this in a few places. And in between them, you have the pleural cavity. What else are you going to call it, right? And within the pleural cavity, there's fluid there, pleural fluid. And there's about 30 milliliters or so, give or take. What do you think the purpose of that fluid is? Well, think about it. What's going on in the lung? You got expansion, contractions, expansion, contractions. There's a lot of movement. And you have these 2 layers surrounding it. So it would make sense you'd have something in between for lubrication. And that's what it's there for. It's there to kind of, again, break down some of the tension, allow for that expansion to happen without any kind of tearing, right? If you didn't have that fluid in there, then there would be a lot more friction, a lot more resistance to breathing. It would take a lot longer, right? A lot more energy. So this basically, serves to allow that. So like WD-40, it allows it to lubricate, right? Okay. Hope you're with me still. So ventilation in the membrane, or ventilation in the lungs, pulmonary ventilation. There's one law that really basically drives the whole thing. And it's not quite what people think, so we'll talk about it. But that law is Boyle's law. And Boyle's law is a very straightforward thing. As the volume increases, pressure drops. As volume decreases, pressure increases. And this is the case for anything that can't escape. So in a confined surface, you can manipulate volume and pressure you can manipulate pressure by manipulating volume, right? And this is basically the law that governs ventilation and how your lungs work. So Boyle's law is defined as that. It's the inverse relationship between volume and pressure. And how can you change the pressure within the lungs? How can you make it go up? How can you make it go down? Well, you have to change the volume. Lucky for you, you have all sorts of ways to do that. So the first thing to remember is the lungs are open to the outside environment. So the outside environment is going to have its own pressure. At sea level, we have millimeters mercury, so 760 millimeters mercury is the pressure, the atmospheric pressure of air. Right now we're talking oxygen here. We're just talking air, because all we're doing is moving air. We're not talking about oxygen yet. And then you have your lungs encased by muscle, right? So you have the diaphragm on the bottom end, which is a primary mover. We'll talk about them. And then you have the rib cage, and in between the ribs, you have the intercostal muscles. And those intercostal muscles, along with the diaphragm, are the main muscles powering respiration or powering ventilation. So yeah, I do it too. Now, within the lungs, you have other pressures. You have your intra-alveolar pressure, right? And depending on the volume, that pressure will change. And you also have a pressure that's exerted in the pleural cavity, or the intrapleural pressure, right? Which tends to stay constant and tends to be a little lower. It has to be a little lower than the intraalveolar pressure. And the reason is because it's there to provide a little support and a little lubrication, but it's also there to basically not hinder the expansion of the lungs, right? Now, if for whatever reason that intrapleural pressure increases, well, that means that there's only a limited space in there. So that means intra-alveolar pressure is going to decrease. So for example, it's the differences in pressure between these two, or what we call the transpulmonary pressure, that's going to drive inspiration and expiration. This is important. because sometimes what will happen is people will tear, they'll damage that part, and they might end up having a wound, like a bullet wound. For example, I remember this one movie, Three Kings. It was about the first Gulf War, and this one guy got a bullet wound that was basically causing his he couldn't breathe because the pleural cavity was penetrated and air was rushing in, and it basically took up the volume that the lungs would normally breathe, right? And they actually did a pretty good job of explaining this. They even had a graphic in the movie. One of the few good Mark Wahlberg movies, and Ice Cube was in it. One of the few good Ice Cube movies that wasn't Friday, the first Friday. So this is important because the differences in pressure between the inside of the lungs and the outside atmosphere is what's going to drive inspiration. Now, here's the thing. A lot of people believe that the diaphragm somehow contracts and forces air out. That is the opposite of what happens. The diaphragm is usually in a higher up position, in a superior position. And when it contracts, it flattens out. And by flattening out, it opens up room for the lungs to expand in their volume. How do they expand? Well, the partial pressure of air inside the intra-alveolar partial pressure, the partial pressure inside the lungs, is lower than that of the atmosphere. So the way you expand is just by opening your mouth or having access to your nostrils. Air will naturally rush in to fill up the lungs. So by contracting, all the diaphragm is doing is providing space. The lungs expand, fresh air is pulled in, inspiration, gas exchange happens, and then when the diaphragm contracts, well, the lungs are basically, the lungs basically shed the air and expiration. So it is the ability of the diaphragm to contract and provide space that drives inspiration, right? And then when it relaxes, the volume decreases, air is forced out because pressure has built up now. So on the next breath, stuff's moving out. And that's how it works, right? So regular breathing, or eupnea, right? E-U-P-N-E-A. P-N-E-A just refers to anything having to do with breathing. That type of regular breathing, it involves your diaphragm. At the same time, when your intercostal muscles contract, what they do is they open up the rib cage slightly, and all of that together drives inspiration. So that's normal breathing. And then depending on the situation, you can over-rely on certain muscles than others. So if you're doing shallow breathing, then most of the breathing is done by small little contractions of the intercostal muscles, which open up the rib cage a little bit. It's very quick to respond. If you take a deep breath, it's diaphragmatic breathing. You're basically relying a lot on your diaphragm, and you're basically maximizing the space to bring in a deep breath, right? Now, if you are, needing to breathe in a lot more, forced breathing, hyperpnea, singing, exercise, well, diaphragm and intercostals are going to work to their maximum, but you can also rely on other core muscles to actually help provide even more space and more stabilization to allow you to do, a bigger breath, right? So, all these accessory muscles are also involved in an indirect kind of way, all right? So like singing, for example, you're forcing air through your voice box and you're doing it at high rates sometimes. That requires, sustaining a note. That requires a lot of muscles to do that, right? So you don't think of singing as this activity that requires muscles, but it does. So these are all different types of breathing and the roles that the different muscles play. So let's talk about birds, because birds they're the shit when it comes to breathing. So ventilation in birds involves air sacs, and there could be anywhere from 8 to 11. And how it works is every time air gets brought in, right, during a breath, they're breathing in, that air rushes to the back and fills up these air sacs. Abdominal air sacs, caudal air sacs is filling up these air sacs. Nothing's happening to these air sacs. They're just holding onto the air temporarily. All this happening in one direction. And then at the same time, air that was in the lungs that has been now depleted of oxygen is now going to move its way out of the lungs into another set of air sacs near the front of the bird, right? So your intraclavicular, which is located kind of, the wishbone of a bird, kind of in between there, and other sort of ones that are kind of closer to the opening, if you will, right? Thoracic air sacs. So that stuff's going to be depleted, deoxygenated air that's already been extracted of oxygen, right? So it's shown here in blue, whereas the fresh air that's come in, that's shown in red, right? The oxygen-rich air doesn't have a lot of carbon dioxide. The blue air has a lot of carbon dioxide. On the next breath, what's going to happen is that blue air is going to be expelled and fresh air is going to be brought back in. And during that time, while the blue air is filling up the air sacs and is diffusing out of the lung, the air sacs that are in the back are slowly releasing, well, not slowly, pretty quickly releasing their oxygen-rich air into the lung. So what this means is that your lungs do not have the problem that we have with our tidal lung. Because of these air sacs, birds don't have a residual dead space. They just have a constant movement of oxygen-rich air. There is no dead space. There is no taking the partial pressure of oxygen and that's really high brought in from the outside environment and then mixed with stale air that just happens to be in your lungs anyway. Right? They don't have that problem. They just keep it moving, basically, literally keeping it moving. And that has allowed birds to have this high metabolism and be capable of these incredible things like migration and stuff like that. It's in part because of this highly efficient ventilation/respiratory system. And this is coupled with other adaptations that we'll talk about in the future. So quite amazing. Far cry from that sea cucumber, I'll tell you that. Okay, so now let's talk about some of these other things that are important. So organisms, animals have respiratory pigments. And what respiratory pigments tend to be is they tend to be proteins that have some sort of metal attached to them, so metalloproteins. And they tend to be fairly large conjugate molecules. So what they do is, I'm sure you know, we've probably heard of hemoglobin, right? Hemoglobin has iron. It has 4 of them, right? It's the every time we talk about protein structure and we talk about quaternary structure, we're almost always talking about hemoglobin. So you have 4 of these different heme groups that have attached to each other, and they have a central part. So the central heme group is the iron, and it's that iron that's able to pick up oxygen, right? So that's the role of respiratory pigments. They're picking up oxygen or picking up something else. So quick thing, what is the difference between being oxygenated and being oxidized? And what's going on with these pigments? Are they being oxygenated or are they being oxidized? And what's the difference? Well, it's a pretty, they look similar, but they're pretty different. Oxygenated is what's happening here. So an oxygen molecule is binding to the iron, and it's a reversible thing, luckily, and it's basically a process involving a covalent bonding. Oxidize or oxidation occurs when you have, as you know, it's a redox, right? So it's occurring when you lose an electron, or you lose an electron, or if you're in water, it's going to look like losing a hydrogen. But the bottom line is it's not necessarily a covalent bond that's forming between something. And it's reversible because you can have a redox, right? You can oxidize, reduce. But they're not the same thing because they involve movement of electrons as opposed to actual binding of oxygens. So we're talking about respiratory pigments, we're talking about oxygenation and binding of oxygen. So hemoglobin is the one that vertebrates have. Obviously, it takes on a reddish hue when oxygen is bound. When it's not bound, it takes on a slightly darker hue. Sometimes it's shown as blue, not really blue, but anyway. Another thing we have is myoglobin. So in our muscles, we have myoglobin that instead of having 4 irons attached to it, it's basically just a single unit of iron, but it again binds oxygen. If you start looking in invertebrates, you get more variety. You get a number of different things that use either varying forms of hemorrhinin or chlorocrutin, which is a lot of different marine worms have these, and they can have a number of them. There's some out there that have binding sites for 12 oxygen molecules, right? Because they have 12 irons attached to them. And then I already mentioned hemolymph. Hemolymph is greenish in color because it has copper, and that is bound to hemocyanin. And we already mentioned how that copper is not necessarily involved in oxygen and respiration, but we still use it anyway. Quick thing, the blue blood from horseshoe crabs. This is especially important for vaccines. What they'll do is they'll isolate a protein that essentially responds to bacterial infection. And what they'll use these is for quality control for vaccines for shelf life. So they'll know that a vaccine is okay by having this sort of indicator. And it's a very costly thing for the horseshoe crabs, but they do have these sort of facilities where they basically harvest the blue blood from these guys, and the blue blood comes from the hemocyanin, right? So it's an example of sort of being able to find a biomedical use for something that's in nature. Right? For the most part, they have to devote resources. Companies that do this have to devote resources to preserving horseshoe crabs. So it's kind of a good thing, I guess. So they drive these partial pressure differences and allow for diffusion to happen in a sort of artificial way. So this is a schematic showing the tension, or tension is just a fancy term for partial pressure of oxygen. High tension, high partial pressure; low tension, low partial pressure. So across a barrier here in this first indicator, in this first situation, you see there's no hemoglobin involved. You have gas diffusion, and it's going to do what it does. It can go wherever it wants, so it's going to set up an equilibrium. No big difference. Now in the second one, the one in the middle, you now added some hemoglobin. So what's hemoglobin going to do? Well, it's going to bind up some of these oxygens, and by binding up these oxygens, it basically prevents them from going to the other side. and it starts to establish a new gradient. So you see here, even though there's the same number of white circles on either side of the barrier, when you look at the ones that have the arrow, there's fewer of them. So now you're establishing a new concentration gradient, differences in oxygen tension on either side. So now the oxygen molecules on the A side, they're going to naturally, through diffusion, go towards the B side to establish a new gradient. And that new gradient gets established in the third picture, right? So you have this new equilibrium that's formed. So now you have oxygen molecules, but most of them are in B, right? Even though the free oxygen molecules have established an equilibrium, you've still taken these other ones. So you have oxygen now in your blood. So A would represent the air in the lungs, and B would represent blood, right? So now you're able to pick up the oxygen, right? And that's pretty cool because you're also doing what I told you oxygen doesn't want to do, which is that you took it out of the air and put it into the water where it doesn't want to be, right? It prefers to be in the air, right? So you've taken it out of the gaseous stage and forced it into solution, basically. Quick word on myoglobin. Myoglobin, as the name suggests, myo means muscle. So it's basically like, it's kind of like hemoglobin, except that it's only 1 as opposed to 4 iron groups. So it's monomeric. It does tend to have a slightly higher oxygen affinity than hemoglobin, and it allows you to keep oxygen right at the muscles where they need to be, right? So muscles are a place that have a very high oxygen demand, makes sense. And you also have a lot of mitochondria, makes sense. So they're going to be keeping a reservoir, basically, of oxygen right by the muscles for that use, right? So it's just one more adaptation to dealing with that, right? And unlike hemoglobin, which has more of an S-shaped curve, because there's a single binding site, boom, oxygen has a typical hyperbolic curve, saturates fairly quickly when oxygen is present, again, because it has a higher affinity than hemoglobin does. But hemoglobin's curve is a little more like an S. So this is showing the saturation of hemoglobin. Again, there's 4 binding sites. And then it's showing differences in oxygen tension in the veins, so systemic venous partial pressure versus systemic arterial partial pressure. So notice there's a difference, right? Oxygen in the arteries has a higher tension of oxygen than in the veins, makes sense. When you follow the curve, however, from the arterial and the venous, that difference in hemoglobin saturation, what do you think that amount represents? Well, that amounts about, let's say, 23-24% of the total oxygen that's available. And that's the amount that gets unloaded by your respiratory system to your tissues, because the arteries are taking blood away from your heart, veins are bringing blood back to your heart. So that vein blood coming back is the stuff that's been used by your body. So right off the bat, it tells you that the capillaries, only about a quarter of the oxygen molecules that you're bringing into your body actually end up in the tissues. So that's about how efficient we are. 25% efficient, which I would add, if you had a person that was an employee of yours that was only 25% efficient, you would probably fire that person. But for respiration, it seems to be okay. Now this curve, this oxygen hemoglobin dissociation curve, can shift. And specifically, it can shift in response to carbon dioxide, which we haven't really talked a lot about carbon dioxide yet. So this curve here on the right is showing oxygen content of blood as a function of partial pressure of oxygen. And you notice that there's 2 curves. There's a straight line, which is dissolved oxygen, okay? But then there's the curve. So that's showing hemoglobin binding up oxygen. Well, basically, the binding of hemoglobin to oxygen, the affinity to bind oxygen by hemoglobin, can be influenced by pH. Lower the pH, right? And lower pH means more acidic. So the more acidic blood becomes, the lower the affinity for oxygen, right? And then vice versa. So we talk about this shift as being the Bohr shift or the Bohr effect, which is saying that as pH decreases, oxygen affinity decreases, and also the total amount of oxygen that can be carried by hemoglobin decreases. So the shift in affinity is called the Bohr shift or the Bohr effect, and then the decreased ability to hold onto oxygen is called the root effect. And you can see that they're related. So if you look at that graph right there, it shows you how they're related. So the difference between the reddish or the burgundy line, high pH to the low pH, that difference is the root effect. So in low pH blood, less oxygen is being transported. So now you're probably wondering, where does the pH come from? Well, it's got to do with CO2. We'll talk about that. On the flip side, there's another thing. Hemoglobin can also bind carbon dioxide. We don't talk about it much, but it can also bind carbon dioxide. So in a lot of ways, carbon dioxide is a competitor to oxygen for the binding site on hemoglobin, for that heme group. But normally, oxygen is the preferred ligand. So when there's high amounts of oxygen available, CO2 barely binds. There's very low affinity for hemoglobin for CO2. But as the partial pressure decreases of oxygen, well, CO2 affinity increases. So we call this the Haldane effect. So this is showing the other graph here where it shows 3 different partial pressures of oxygen. So where there's high amounts of oxygen, the total blood CO2 decreases. All right? So this relationship between CO2 and O2 is called the Haldane effect. So you see these effects in particular places, and they're absolutely vital for respiration. So you see them in places. So where do you think you see these differences? Where high amounts of oxygen are present and high amounts of CO2 are present, where they influence each other's dynamics? Well, this is the pièce de résistance right here. This is how respiration happens. When you start with a cell that's used up oxygen and has produced CO2 as a product of respiration, that CO2 likes being in solution, so it dissolves. And then as it goes through the interstitial fluid and enters into the bloodstream, a CO2 molecule will have one of 3 fates. It'll either just stay as a dissolved CO2 and just kind of enter into the plasma of the blood, right? So that'll represent about 10% or so of the CO2 molecules. Then another 20 to 30, 25 to 30% of that CO2 is just going to bind to hemoglobin, right? So again, we're at the tissues. So a lot of the hemoglobin had oxygens, but they gave up the oxygens. And in their place, they got the CO2. So when that happens, you produce basically a hemoglobin that has a CO2 group attached to it. But the majority of CO2 is just going to react with water. CO2 plus H2O in the presence of something called carbonic anhydrase, right, the enzyme. What it's going to do is it's going to produce carbonic acid, H2CO3, and it's an acid. And what do acids do? Well, acids dissociate. It'll dissociate into a bicarbonate ion, but it'll also dissociate into a proton. And that dissociation of the acid into a proton, that's going to drive down pH. It's going to lower pH. Now, H2CO3 breaks down into a proton and a bicarbonate. Bicarbonate is HCO3 negative ion, right? Anion. So that bicarbonate tends to move out of the cells. So another thing I'll mention is that red blood cells have to maintain their cellular integrity, their electrochemical gradient. Since they're losing this negatively charged ion a lot, what they'll do is they'll import chloride shift it's called. They'll import them into the cell just to maintain that electrochemical gradient that they need to function. So the bottom line is CO2 enters into the blood and starts to lower the pH. If you lower the pH in and around the tissues where there's a lot of CO2 production, well, that's not very conducive to hemoglobin holding onto oxygen. So what does it do? It lets it go, right? Lowered pH, metabolic acidosis, which happens, this is what it's called, metabolic acidosis. Lowering of the pH makes hemoglobin release oxygen right where it needs to release it, right where the CO2 is being produced and oxygen is being used up. It's a great system, right? So again, it's that Bohr shift, that root effect. It's causing hemoglobin to just release oxygen, and you're sweet, right? You're releasing it right where you need to because of the local pH changes. On the flip side, you now have hemoglobin moving around with CO2. You have this carbonic acid floating around and CO2 that's dissolved. Now you're making your way through the circulatory system and you're picking up CO2 as you go, dropping off oxygen as you go. Eventually you make your way back to the lungs. So now what's happening? Well, now we're at the lungs. The lungs are a high O2 environment. High O2 environment, high tension. So what's going to happen? Well, you have hemoglobin. So now that it's a high O2 environment, you're going to get oxygen coming into the bloodstream. In the presence of high oxygen, you have CO2 still attached to the hemoglobins. Well, that affinity decreases with high amounts of oxygen, i.e. the Haldane effect, right? So we saw the Bohr shift and the root effect taking place at the tissue level, but now at the lungs, you're starting to see the effects of the Haldane effect. So because of the Haldane effect, high amounts of O2 decreases affinity of hemoglobin for CO2. CO2 bounces, takes off, gets released off from the hemoglobin, and because there's this gradient, right, with very low amounts of CO2 outside, the environment favors that CO2 leaving. Dissolved CO2 at the same time, it maintains such a high gradient that would actually rather be in the air where it doesn't want to be than be in solution with oxygen. Like they hate each other, right? They hate each other. It's like pro wrestling, like they hate each other like that, right? Like they can't stand to be in the same place. So now CO2 is going to leave and enter into the lungs to be expelled. That's the dissolved CO2 that's going to happen, right? At the same time, you had the acids over there, the carbonic acid. Well, that reaction is going to basically reverse, and it's going to produce, it's going to allow CO2 in its dissolved form, right? So it's going to react, basically the opposite of that carbonic anhydrase driven reaction. That's going to happen. Hemoglobin is going to release its CO2. There's a lot of CO2 there, but it doesn't want to be there because there's a lot of oxygen there. Oxygen's taking its place in the hemoglobin. So now, because of the Haldane effect, it's going to force CO2 out of solution into the air inside the lungs to then be expelled. So beautiful system, right? Bohr shift, root effect, working at the tissues. Haldane effect, working at the lungs. Beautiful. I think it's beautiful. So, to get back to our original question about the differences in respiration between water and air, well, they present different challenges with regards to getting oxygen in the first place out of the environment, extracting it, and releasing the CO2. And releasing CO2 especially is important because that can shut things down. Your body is really trained to respond to that metabolic acidosis. So whenever you hold your breath and you force air, you eventually have to take a breath, it's because of CO2. If you're underwater and you're holding your breath and you can't escape, eventually you're going to have to open your mouth. The reason is because of CO2 buildup. That acidosis forces you to breathe again. Your whole body's attuned to this. So it's unfortunate. We're kind of a slave to CO2, so to speak. So how do animals overcome these issues? Well, they can maximize their respiratory exchange areas, right? Their lungs, their gill surface area, right? They can manipulate partial pressures using pigments and these effects of these different concentrations and pHs and gases and stuff like that. And that allows respiration to occur as this well-oiled machine that we're all thankful that we can do. So that's all for today, and join me next time. Have a good one.